Hello and welcome back to part 2 of this video series on the respiratory system. So let's go ahead and pick up where we left off last time. Next we are going to start discussing spirometry. We spent a lot of time talking about the pressure-volume relationships as it pertains to our ability to mechanically draw air into our lungs and to push air out. So spirometry is all about the actual physiological measurement of the capacity of the respiratory system, especially as it pertains to the volume of air that we can pull in and out. So there are a lot of different terms for us to get acquainted with here in terms of volumes and total lung capacities, in terms of what is physiologically relevant and what is not. But before we get into this, let me go ahead and show you what a typical spirometric setup is going to look like. This is the sort of thing that we would be doing in lab, but obviously because of COVID-19, we cannot. So let me go ahead and show you what this kind of looks like. So essentially, whoever your subject that you are performing spirometric measurements on, your subject is going to be basically breathing into and out of a mouthpiece that is connected via a hose to something that kind of looks like this. And now this is kind of an archaic setup, so these sorts of things can be done more of a technologically advanced way these days with computers and such. But this is kind of what a archaic spirometry setup might look like back in the old days. So we're familiar with, obviously, the idea with inspiration and expiration, that as we breathe in and out, we are pulling air into our lungs and pushing air out. So if you are doing spirometry here, the air that you breathe in and out is coming into and out of this hose. And specifically, the air is being drawn into and out of this big chamber right here with water on either side in these two reservoirs. Now, the air that is changing in and out of this chamber right here is going to affect the position of this kind of bell right here. You might think of it as kind of a weight that kind of shifts up and down as the amount of air that's coming into and out of this chamber changes. This bell is connected via a pulley system that is connected to a writing device that is going to track up and down as you breathe in and out onto this drum that spins around. And because the pen is being put to the paper on the drum, we can see as we breathe in and out. So if you take a closer look here, you can see an inspiration, the pen goes up, an expiration, the pen goes down. And that's all because the amount of air in this chamber continues to fluctuate as we pull air in and out of our lungs. So if we back up here, we're going to go through all of these different terms here as we go through, but basically what I'm going to do here is I'm going to walk you through what a typical spirometric procedure is going to look like. So the first thing your subject is going to be instructed to do is to just breathe as they normally would. So you don't have to have your subject do anything special. They're just going to breathe in, breathe out normally as they typically would. So what you are measuring here is what is called the tidal volume. The tidal volume referring to the volume of air that is drawn into and out of your lungs with every restful inspiration and expiration. So after you get a couple of tidal volumes, next your subject is going to be asked to breathe inward very forcefully. So basically what you're telling your subject is suck in as much air as you possibly can. So essentially, breathe in as much as you possibly can. So what that's going to do is it's going to cause the spirometry graph to go up and up and up and up until it can't go up any further. What you are measuring here is what is called the inspiratory reserve volume. So essentially, on top of the normal tidal volume of air that you draw into your lungs, how much extra space is there in your lungs? How much extra air could we fit in there theoretically? So next, after you do that, you're going to exhale forcefully. So you draw in as much air as you possibly can, breathe it out. So now you're back to just a regular tidal volume. Now your subject is going to be instructed to breathe out as much as you possibly can. So basically, the idea that your subject should have is basically try to empty your lungs. Try to breathe out as much air as you possibly can. So that is going to cause the spirometry graph to start to take a nosedive. So you can kind of see that right here. So you can see we go down and down and down. So this is going to give you your expiratory reserve volume. So how much air is stored up in your lungs that you are actually capable of breathing out on top of the tidal volume that we mentioned before. Now, what you're going to notice on this graph here, if you look at the y-axis here in terms of total cubic centimeters or milliliters of air that can fit into your lungs, you'll notice that nowhere on this spirometric graph do we actually get down to zero. Nowhere on this graph do we actually get to the point where there is no air in the lungs. And that is because it is not possible, physiologically speaking, to completely empty your lungs of air. That is because of what is called the residual volume. So the residual volume is the volume of air that is always in your lungs no matter what. It is basically the minimum amount of air that needs to be in your lungs to keep them inflated and to keep them from collapsing. So obviously if you completely empty your lungs of air, the lungs are going to collapse on themselves like a vacuum. So basically the way to think about this is the residual volume is air that can never be forcefully expelled from the lungs under physiological conditions. So because that air is always there anyway, the residual volume does not play any sort of functional role in increasing our respiratory capacity or anything like that. If, say, you're exercising, the residual volume doesn't really have anything to do with you getting extra oxygen into your blood or anything like that. So when you are talking about the amount of air volume that can be put to functional work for you in terms of extra oxygen, getting extra carbon dioxide out, that is what's called the vital capacity. The vital capacity runs, you can see here, from the bottom of your expiratory reserve volume to the top of your inspiratory reserve volume. So basically, your vital capacity is the total lung volume, meaning when you fully inflate your lungs by breathing in as much as you possibly can, your vital capacity is that total volume minus the residual volume, because that volume of air is always there anyway. So your total lung capacity in this case would be almost 6 liters, and then you would have to subtract a little over 1 liter to actually get your vital capacity, the actual volume of the lungs at any given time that is actually capable of being put to work for gas exchange. So a question that I want you guys to think about for our Collaborate meeting this week: of the parameters that we've talked about, things like tidal volume, reserve volumes, total lung capacity, etc., which one of those do you think is going to increase with exercise? So in order to help you determine the answer to this question, you need to make sure you understand the strict definitions we put on those different terms, and then also imagine yourself actually having your spirometric parameters measured. Imagine what your graph would look like if you have just been exercising. So you're going to be breathing kind of heavily. So imagine what your spirometry graph might look like after you've been exercising, and we can discuss the answer to this question on Wednesday or Thursday, depending on which class you're in. So a common test that is done with spirometry, so spirometry is not something that's done just for kicks and giggles, so the whole purpose of spirometry is to measure the capacity of someone's respiratory system, and in specific cases we can use it to diagnose either restrictive or obstructive disorders of the respiratory system, things like asthma, pulmonary fibrosis, or emphysema. So if you're dealing with something that is restrictive, we're talking about something that has narrowed the airway. So maybe you've got too much parasympathetic input like we talked about before. The airways have narrowed due to bronchoconstriction. So if that is the case, air is not going to be moving out of the lungs as efficiently as you might expect, and that should show up on spirometry. Or if you're dealing with something obstructive, then some foreign material, whether it's something you've breathed in, or maybe it's some sort of cellular or fluid material that is accumulating in your airways, something that is actually blocking the airways rather than restricting it, that really the same way should slow down the efficient movement of air from the lungs out and back in. So these sorts of things should be readily visible on a spirometric graph. So one very common test that we can use to determine this is what is called a forced expiratory volume in 1 second test, or an FEV1 test. So basically the way this test is performed is that and this chart's a little tough to read unless you know to read from right to left. So if we start over here on the right, you'll notice that this subject, as they breathe in and out of the hose, they're being instructed to just breathe normally. So what we're seeing here is their tidal volume, which should look pretty familiar by now. So you'll notice that after a few tidal volumes, our subject is instructed to get your inspiratory reserve volume. So they breathe in as much as they possibly can until they can't breathe in anymore, and then they hold it for a minute. Not a minute, but they hold it for just a second or two. So then, when they're ready, they're instructed to breathe out as much as they possibly can. So again, now we're going to the expiratory reserve volume. So we've already seen how this works in the previous graph, but here is what we're keeping track of here. What we're looking for here is not necessarily how much volume can you push out total. We're looking at how quickly can you push it out. Specifically, we are keeping our eye on how much volume of air can you expel from a fully inflated lung in exactly 1 second. So in this red graph right here, you'll notice that we go from a maximum lung volume of, say, 5 liters, and after 1 second, we are down to 1.8 liters. So in 1 second, we are able to expel 3.2 liters of gas, roughly 60 to 70% of the total lung volume. Okay, but look at this purple graph right here. You will notice that eventually the patient that's producing the purple graph, eventually they get down to the same level of expiration as the patient in the red graph, but after 1 second, they only go from 5 to 2.5. So as opposed to 60 to 70% of the lung volume, they're only able to get out about 50%. So just from looking at these graphs, we can diagnose the patient that has the purple graph with some type of disorder that is preventing the speed or the efficiency of air moving in and out of the lungs. Because there's either something restricting or obstructing the airways, the air cannot move very quickly, which is why it takes a little bit longer to go from 5 all the way down to your maximum expiration of 1 liter, which again is your residual capacity. And then there are a couple of other types of spirometric measures that can be done, and you can see some of them here. So peak expiratory flow, a couple of them we've already looked at here. So you only really need to keep track of the ones that we have already discussed, but there are a couple of other ones that are kind of interesting and can be measured. So you can take a look at that if you want. Now, it is worth mentioning before we move on that spirometry, really all it's capable of measuring is specific lung volumes. So what we are about to start talking about in terms of neural control of breathing is that your central nervous system helps to regulate your breathing rhythms based on specific chemical values like the amount of oxygen and carbon dioxide in the blood itself. So those are not things that can be measured with spirometry. Those would require a specific type of chemical analyzer that actually measures these things in the blood. Okay, so speaking of neural control of breathing, we've already talked about how all of those thoracic muscles, including the diaphragm and the intercostals, are going to contract and relax in order to regulate and change the volume of the thoracic cavity in order for us to change the volume of the lungs, change the pressure, and to get air coming in and out. So what I'm sure you are aware of is that because all of these different thoracic muscles are skeletal muscles, they're under the control of somatic motor neurons, meaning that we have a level of voluntary control over these. So anytime you voluntarily want to take big, deep breaths, even if you don't need to, you can do that. You can start huffing and puffing air anytime you want. And that is because these things are under the control of somatic motor neurons. So here is a review question for you to kind of do on your own, going back to some of our Unit 3 stuff. So I recommend you go ahead and do that just to kind of refresh yourselves. So the thing to understand, though, is that obviously we don't have to consciously focus on breathing all the time. This is not necessarily something that is only voluntary. It turns out that the control of these thoracic muscles is actually under some level of autonomic control as well. So we've talked about autonomic control centers plenty of times in the past, so you will recall that there are respiratory control centers located both in the medulla and in the pons. So a lot of our autonomic control of breathing comes via spinal nerves. So essentially you can picture pathways leading either from the brainstem, the pons, or the medulla, down through descending tracts through the spinal cord, and then out through the appropriate spinal nerves. And the targets for those spinal nerves are obviously going to be the thoracic muscles. So our central nervous system is capable of regulating our breathing because those somatic motor neurons have actual control. They actually innervate the skeletal muscles of our thoracic cavity. So we've already looked at some autonomic control functions as it pertains to things like blood pressure and heart function. So we've already looked at how when things like your blood pressure change, whether it's an up change or a down change, the autonomic nervous system is capable of communicating with either the SA node, the AV node, or the ventricular myocardium in order to change your cardiac output, to change your blood pressure, right? So we are going to look at a very, very, very similar pathway here with just a couple of notable differences, including what is your sensor and what is your eventual effector. Everything else should be pretty similar here, though. So as the case was with blood pressure and heart function, we have these control centers, these autonomic control centers. Again, they're located in basically the same place as before, the pons and the medulla. So our idea here in terms of maintaining homeostasis, which that's been our persistent topic throughout the semester, that's what we're always trying to accomplish. What we are trying to maintain here is we are trying to maintain consistent and adequate levels of oxygen in the tissues, because obviously that is the whole purpose of the respiratory system. We breathe in, we get oxygen into the blood so that we can nourish all of those tissues that are trying to make ATP through cellular respiration. The other thing we are trying to accomplish is we do not want carbon dioxide levels to get too high. Carbon dioxide being that byproduct that is created as a consequence of cellular respiration. We need to get some of that out of the blood as well. So our effectors in these homeostasis loop, don't forget the way we define an effector, as always, is this is the thing that is most directly responsible for changing whatever needs to be changing. So in this case, our effectors are actually going to be the thoracic muscles that produce the volume changes. So there are more neural control centers as far as breathing goes rather than what we've just talked about. So we are going to focus on the ones that are highlighted with these green arrowheads right here. So we've already talked about the respiratory control center. That says Renter right there, so that's kind of an embarrassing typo, but at least it wasn't my fault this time. This is actually in the OpenStax book. Yikes. So we have these two right here. These will look familiar. So we have sensors located both in the aorta and the carotid. You'll remember before we talked about baroreceptors located there for measuring blood pressure. We have different types of sensors here that we'll talk about in just a second, but it is kind of nice for you that these sensors are going to be largely located in the same place. And then we'll also talk about the cerebral cortex too. So we're going to kind of get into the kind of subtle difference between your voluntary breathing, so when you choose to breathe in and out, and when it just kind of automatically happens. So if it automatically happens, those efferent signals are essentially going to be coming from the pons and the medulla. Whereas if you're talking about voluntary controlling, those sorts of efferent signals are going to originate in the cerebral cortex, as we've talked about before. Okay, so as long as we're talking about a homeostasis loop, let's go back to our definition of your stimulus. So your stimulus, the thing that always starts a homeostasis loop, what I have always told you is that it is going to be some parameter that is either too high or too low. It's either too high above its normal range or it's too far below its normal range too. So the parameters that we are going to keep our eye on here, it's not just one thing. It's not just blood glucose. It's not just blood pressure. We actually have 3 of them. The rate of breathing that your medulla and pons are going to respond to are 3 things. The pH of your blood, so how acidic is it, how basic is it. The Po2, so the oxygen saturation in the blood, and the PCO2, the carbon dioxide saturation in the blood. So 3 different things that our respiratory system needs to be able to make changes in order to keep those 3 things within the normal range. So our sensors in this case are called chemoreceptors. Now, they're not baroreceptors as they were for blood pressure. Like I said, it's easy to forget that because we already talked about a baroreceptor reflex in which pressure receptors or baroreceptors in the aorta and carotid measure blood flow and allow us to respond to momentary drops or increases in the blood pressure. It's easy to get confused because we're still talking about the same place, but these are not the same types of receptors as before. These are chemoreceptors. These are the types of receptors that actually respond to chemicals that are dissolved in some liquid medium, kind of like what we talked about with taste and smell. So these chemoreceptors can respond to oxygen in the blood, carbon dioxide in the blood, and hydrogen ions in the blood, which again is what produces changes in pH. So these chemoreceptors form, again, as always, the sensory nerve endings to sensory neurons, and you can see these afferent pathways as they lead from the aorta and the carotid into the brainstem, into the pons and the medulla, and usually they're going to go through one of 2 different pathways, either through the vagus nerve or through the glossopharyngeal nerve, one of the 2. So let's hold off on talking about pH for the time being. It's going to require a little bit of an explanation for you to really understand how the blood pH acts as a signal for how you need to alter your respiration. Oxygen and carbon dioxide we can at least kind of understand right away. So you can understand, I think, that if you are not breathing enough, if you're not getting enough oxygen into your body, you might expect your Po2 numbers to be a little bit low. You're not breathing in enough, you're not getting enough oxygen. As a consequence, if you're not breathing enough, you're not getting enough carbon dioxide out. So you might understand that might cause your PCO2 to be a little bit high. So just for those 2, you can hopefully kind of picture how one being high and the other being low can act as a trigger telling you to either breathe more or breathe less, depending on which is high and which is low. So I mentioned kind of the subtle difference between this voluntary breathing that we talked about before. As I said, those efferent signals are going to originate in the cerebral cortex, but if you're talking about your more autonomic breathing, the sorts of kind of second to second, minute to minute alterations to your breathing rate that you perform in accordance with Po2, PCO2, and pH, those efferent signals are going to originate in the pons or the medulla. So if you look here, we've already said that any small alterations we need to make to our breathing rhythm come initially from chemoreceptors. So we measure the amount of oxygen and carbon dioxide and hydrogen content in the blood. We report those afferent signals to the medulla, and then the medulla will coordinate a response, send a efferent message down through the spinal cord, and then those motor neurons will eventually innervate the thoracic muscles, and we can either get them to contract more frequently or less frequently, depending on whether we need to breathe more or breathe less. So here's something that's kind of interesting, an interesting case study to really help you understand this difference between your voluntary breathing, basically you just up and decide, even if you don't need to, I'm going to start huffing and puffing because that's what I feel like doing. I don't need to do it, but I'm going to do it. And the difference between that and your autonomic breathing. So this disease that we're going to talk about here called central hypoventilation syndrome, which is more frequently called Ondines Curse, is really going to help us understand this difference. So a person with this disease has some sort of defect. It's not completely well understood at this point, but these people have certain defects in the pathway of information, efferent information that leads from your respiratory control centers into the thoracic muscles. So some disruption of that flow of information basically means that people with Ondines Curse, they can breathe, but they can only breathe voluntarily. The only way they can get that information to the thoracic muscles in order to initiate those contractions is if those efferent messages start in the cerebral cortex. They cannot begin those messages here. So think about this here for a second. During your day, when is it most important that you are able to breathe automatically? When is it most important that you be able to breathe in a way that is not voluntarily? And I'm sure you are thinking exactly the correct answer here. That is when you are sleeping, right? When you are sleeping, you don't have conscious and voluntary control over your thoracic muscles, so you really need your central control centers in the medulla and the pons to constantly be measuring oxygen and carbon dioxide and sending those messages to your thoracic muscles to get the breathing to happen on its own. So someone with Ondines Curse will enter respiratory arrest when they fall asleep. So basically, without any sort of treatment, someone with Ondines Curse cannot live if they ever have to go to sleep. All right, so delving further into this topic, we've already discussed how we have 3 different parameters that your central nervous system is constantly going to be keeping its eye on. PO2, which is the amount of oxygen gas that is dissolved in the blood plasma. And don't forget, dating back to chapter 18, we said that PO2 does not actually measure oxygen that is carried by hemoglobin. Your PO2, it is a very good measure of how much oxygen, basically the oxygenation level of the blood, but don't forget it does not refer to oxygen that is carried by hemoglobin. It only refers to how much oxygen is purely dissolved in the blood plasma itself. The PCO2, which is similar, how much carbon dioxide is dissolved in the blood plasma, and then the pH of the blood, which, if you'll recall back to chapter 18, has a set point of about 7.4, so just a little bit basic. So the first thing for us to look at here is what would happen to the levels of carbon dioxide as you transition from normal ventilation to hyperventilation. Let's say that for some reason you just start breathing way too much. Even though you don't need to, you basically just start breathing in and out way too fast. So what you are doing there is you are bringing way too much oxygen in and you are getting too much carbon dioxide out. So the set point for carbon dioxide that we usually set is about 40 millimeters mercury as a PCO2 value. If you start hyperventilating, this number is going to be essentially slashed in half, so it goes down to about 20 millimeters mercury. Now, you might be wondering, okay, carbon dioxide, it's just a waste product, right? What do we care if we don't have as much carbon dioxide in the blood? Well, we're going to talk about that here in just a few minutes. It actually is very important. And then on the other hand, if you hypoventilate, which I think it's a little easier to understand why that's a big problem, your O2 values are going to plummet and now your CO2 values are going to essentially double. So you go from 40 to 80. If you're hypoventilating, you're not breathing enough. You might be holding your breath or something like that. So you're not getting enough oxygen in. You're keeping way too much CO2 in already, so your PCO2 values start to rise as well. So what we are going to talk about now that we can kind of see how both hyperventilation and hypoventilation cause your PCO2 levels to fluctuate, now we can start talking about the blood pH. So if you look at carbon dioxide that is dissolved within the blood plasma, and keep in mind, as a hydrophobic molecule, it can get into and out of red blood cells. And when it does that, when it gets into red blood cells, it can combine with water via a enzymatic reaction that is catalyzed by an enzyme called carbonic anhydrase. What this enzyme does, that is contained within red blood cells, what this enzyme does is it converts carbon dioxide into something that is actually dissolvable in water. Carbon dioxide is not very soluble in water, obviously because it's hydrophobic. But if it is converted into this carbonic acid, H2CO3, it is now a purely hydrophilic substance. So now it can dissolve within the water. It is ionic. And then it breaks up into a bicarbonate ion and a single hydrogen ion. So every time we do this, every time we convert a carbon dioxide molecule into carbonic acid, the pH of the blood is going to go down because we are increasing the concentration of hydrogen ions. If you don't quite remember the relationship between hydrogen ion concentration and pH, I recommend you go back to the first unit and take a look at that, especially as it pertains to the pH scale and buffer systems. So, for example, if we are hypoventilating, then we are increasing the concentration or saturation level of carbon dioxide in the blood. By mass action, we are going to push this equilibrium towards the right, meaning the more carbon dioxide there is in the blood, the more carbonic acid we are going to create, and the more hydrogen ions are going to be generated, and the pH of the blood is going to start going down and down and down and down. So not only can we measure hypoventilation by looking at how much CO2 has gone up, we can also tell that hypoventilation is probably going on if the pH of the blood starts to go down. Now, what about the opposite scenario? What about hyperventilation? So if you're hyperventilating, then the levels of CO2 in the blood plasma are going to go down. And similarly, as a consequence, there's going to be less carbonic acid and fewer hydrogen ions, meaning the pH of the blood would instead go up and become even more basic than it already was. So we've already talked about these chemoreceptors that are located in the carotid and the aorta, and then also we do have chemoreceptors located within the medulla themselves. So you can actually see this process of converting carbon dioxide and water into carbonic acid and how that hydrogen can actually be directly sensed by these chemoreceptive sensory neurons that are located in the medulla. So we can directly report on the pH status of the blood plasma to those respiratory control centers so that we can alter our respiration to either give ourselves less carbonic acid or more carbonic acid depending on the need of the systemic system. Okay, so oxygen, carbon dioxide, and pH. So these are the three things that we are keeping our eye on. So if I were to, before we go any further here, so let's go ahead and kind of forget everything we're looking at here. So pretend none of this is here right now. Go ahead and develop an opinion on this. Of those three parameters, oxygen, carbon dioxide, and pH, which one do you think is probably the most important for us to keep our eye on? Let's say that of those three parameters, you can only monitor one of them at any given time. Which one of those three would you probably pick? Your inclination is probably to pick oxygen, right? And I'm not saying you're wrong about that. Your instinct is probably pretty good there, right? So oxygen is obviously very important. We need to get it to our tissues, so the whole purpose of the respiratory system relates back to oxygen. So if you pick oxygen as the one thing you want to keep your eye on, I can't blame you. But I'm here to tell you that that is not really the case. If you were going to pick one, oxygen is probably the one you would not want to pick, and here is why. Oxygen is a little bit unique in the sense that not only is it dissolved within the blood plasma, we keep our Po2 set point at about 100 millimeters mercury. Not only is it dissolved within the blood plasma, but we've already mentioned before how the vast majority of our oxygen is carried by hemoglobin in red blood cells. The Po2 value does not account for all of that oxygen. So if your oxygen status starts to kind of fall off a little bit, if you stop breathing, it's going to take a little while in order for you to really start to appreciate your oxygen level falling off. So I have also by now posted a very short review video that kind of demonstrates why this is. So at this point, you might want to pause this video and go take a look at that video. That video shouldn't be very long, so you should be able to kind of knock that out and then come back here. So even if you stop breathing, it's going to take a little while for your oxygen levels to kind of start dropping off. Whereas almost immediately, if you start hyper or hypoventilating, your pH and PCO2 values are really going to start changing. So the whole idea there is that if you want to maintain homeostasis, you want your effector mechanism to activate as soon as humanly possible after the stimulus occurs. If you are using oxygen as your stimulus, you're basically going to be waiting until things get really, really bad until you basically start thinking about hopping into action. Whereas if you're measuring carbon dioxide and pH, you can fix things before your PO2 levels really ever start to change at all, which, if you think about it, is really the ideal way of going about this. So the bottom line here is that our fine-tuning of our breathing rhythms is usually performed in response to elevated carbon dioxide rather than depressed oxygen. If you are not breathing enough, your carbon dioxide levels are going to go up long before your oxygen levels ever start to drop off. So our most sensitive respiratory mechanisms are fine-tuned so that they are performed in response to carbon dioxide changes instead of oxygen. So you can see in this graph kind of how this is true. So what you're looking at here on the Y, this is kind of a complicated looking graph, so I'll try to orient you here. What you're looking at here on the Y-axis is your total minute volume. So what volume of air are you drawing in and out per minute? So basically, how much are you breathing? The higher the graph goes, the more you are breathing. And then on the X-axis, you're looking either at how much carbon dioxide is there or how much oxygen is there. So if you look at the red graph, in which the amount of oxygen is kept normal, meaning oxygen is not changing, we can consider our internal PO2 numbers to be just fine, but carbon dioxide starts to go up, you'll notice that almost immediately after we start getting carbon dioxide a little uncomfortably high, our autonomic control mechanism jumps into effect and we start breathing more and more and more and more. Our minute volume starts to go up and up and up. But on the other hand, if we keep carbon dioxide normal at about 40 millimeters mercury, but we start changing the oxygen content, we start decreasing the oxygen content, you'll see that it takes quite a while for the minute volume to start going up. So this graph here is just another way of showing that really our body is better suited to change our respiration rate in response to carbon dioxide rather than oxygen. Now, that's not to say that oxygen is not important. It absolutely is. But the idea here is that we are so replete with oxygen, both in the blood plasma and in hemoglobin and in red blood cells, that it's going to take basically getting hit with a sledgehammer for oxygen to ever change. So we don't want to wait until things get that bad before we start making changes there. Yeah, and then here you can kind of see a little bit of extra information on things like carbon dioxide, pH, and PO2, where those things are sensed and whatnot. So PO2 is something that is measured, but basically what I've just been trying to say is that it's not the sort of thing that you want to monitor if you're talking about minute-to-minute or second-to-second changes to your breathing rate. So absolutely, you do want information on oxygen when things get really, really bad. Okay, so let's finish things off here in this chapter by talking a little bit more about gas exchange. So this is something that we kind of already covered at the beginning of this chapter. So we know that in the pulmonary capillaries, oxygen-poor blood comes in through the pulmonary arterioles. We get gas exchange to occur. We get oxygen from the alveolar air into the blood plasma, and then we get carbon dioxide from the blood plasma into the alveolar air. And that now oxygen-rich blood and CO2-poor blood returns to the left atrium through the pulmonary veins. So in the review video that I posted the other day, we also covered the process of gas exchange in the systemic capillary, so in the systemic circuit. Here, oxygen-rich blood comes into tissues through arterioles, and after gas exchange, after we deliver our oxygen-poor blood will return to the right atrium through the vena cava. So obviously, we also want to keep our eye on carbon dioxide here. Obviously, if we want to deliver oxygen to tissues, we need to also be able to take carbon dioxide away. And hopefully by now you are pretty much familiar with the idea that cellular respiration in these systemic tissues is what produces all that carbon dioxide that needs to be cleared out. And we've talked obviously about the concept of concentration or pressure gradients that dictate the direction of oxygen and carbon dioxide movement, so we won't talk about that again necessarily. So if you are still a little confused on this, you might look over this slide more carefully, maybe look at that review video again, but I'm going to go ahead and move forward with the assumption that you're pretty good on this. Another thing that we've discussed is that the total atmospheric pressure, if you're at sea level, is approximately 760 millimeters mercury. Here in Oklahoma, it's probably more like 750 or 753 or something like that. So the four most important gases that make up the atmosphere, these are not the only four gases in the atmosphere, but they are the four most relevant to us, I would say. Nitrogen, not because it's physiologically relevant, it's just kind of hard to ignore the gas that makes up almost 80% of the atmosphere. Oxygen, which is 21%. Water vapor, which tends to vary depending on the humidity outside. And then carbon dioxide, which again is not very prevalent in the atmospheric air, but because of its immense importance in physiology, we want to be aware of it as well. So something that you likely remember, or maybe not, but it's okay. So something that you hopefully remember from your chemistry class when you talked about gases is something called Daltons Law. So if you have a mixture of gases, which is what our atmosphere is all around us, the total pressure of that gas in the mixture is equal to the sum of the individual partial pressures. So the way to think about this is that if you've got all this air around you, which is mostly nitrogen and oxygen, if that mixture of gases has a total pressure of 760, then each individual gas is going to have some pressure that adds to that total of 760. And the total pressure of that individual gas is going to be relative to how much of that gas that there is. So if you wanted to tally this up, the total atmospheric pressure should be equal to the pressure due to nitrogen plus the pressure due to oxygen plus the pressure due to water vapor, which again is going to fluctuate a little bit, and then the pressure due to carbon dioxide. So we're ignoring things like argon and radon, things that we know are in the atmosphere. So if you look at this, you can see this inspired air. You can see that the total pressure again is 760. Nitrogen is about 80% of that, so about 600 millimeters mercury. Oxygen about 160 millimeters mercury. And then carbon dioxide is mostly negligible just because we said there's hardly any carbon dioxide, physiologically speaking, in the air that we breathe. But hopefully here you can, based on what is in the alveolar air, meaning air that is in the alveoli before you've even taken a breath, you can appreciate the pressure gradients for both oxygen and carbon dioxide, once again dictating the direction in which these guys like to move during the process of gas exchange. Okay, so just a couple more things to clear up here before we finish up this chapter. So I've kind of briefly mentioned it before, but let's talk a little bit more about those Po2 and PCO2 measurements. So it sounds really odd to talk about, but gases are capable of being dissolved in liquids. They don't dissolve very well because we've already covered how most gases, especially carbon dioxide and oxygen, are very hydrophobic. They don't really like being around water, so we can't dissolve very much of them in liquids, but we can dissolve a little bit of them. So anytime you're talking about the P gas, or Po2 or PCO2, you're really talking about how much of that gas is literally dissolved in the blood plasma. So for Po2, again, this measures just the oxygen saturation, just how much oxygen there is dissolved in the plasma, not how much oxygen is carried by hemoglobin. So This is one of the reasons why we said that PO2 is not something that we really want to respond to right away, because for a healthy person that is breathing just fine and is healthy in pretty much every way, there should be plenty of oxygen both in the blood plasma and attached to hemoglobin. So they should be nearly saturated with oxygen all the time. So, as an example, the oxyhemoglobin content for a healthy person is usually somewhere around 97%, meaning of all the hemoglobin complexes in their whole body, 97% of them are fully loaded with oxygen. And then let us not forget something that we talked about before. A big reason why we're able to get so replete with oxygen, how we can get so much oxygen, do not forget how important the immense surface area of the type 1 alveolar cells are. That allows for very efficient gas exchange to occur, is very important in our ability to get oxygen into the blood very, very quickly. So if the surface area here was significantly less, we might be having a different conversation about our ability to carry oxygen. So in terms of measuring oxygen and carbon dioxide levels in the blood, the best and most consistent way to measure this is to get it out of arteries rather than out of veins. There's kind of a lot of fluctuation if you're getting it out of veins, especially if you're dealing with something like exercise, because certain tissues are going to have produced an especially large amount of carbon dioxide, have used up an especially large amount of oxygen. So really the best place to get these measurements is out of arterial blood before we have actually delivered these things to heavily metabolizing tissues. So the main thing to kind of look at here is the partial pressure. So you might want to kind of try to commit to memory that the set point for PO2 is 100 millimeters mercury and the set point for carbon dioxide is 40. So we can actually see how these numbers kind of change as we transition from the systemic circuit to the pulmonary circuit. So in the systemic circuit, coming from the left side of the heart, we have our parameters at their set points. PO2 is 100, PCO2 is 40. After gas exchange occurs here in the systemic tissues, you can see we have picked up some carbon dioxide, it's gone up to 46, and we have dropped off a significant amount of oxygen. We've gone from 100 to 40. But you'll notice that as we make our way back into the pulmonary circuit, we get these things back to where they should be. In the pulmonary capillaries, we bring in more oxygen to go from 40 back to 100. We drop off carbon dioxide to go from 46 back to 40. So that works out perfectly. So internal versus external respiration. This is probably a new concept for you. So let's talk about this. We talked a little bit about it in chapter 18, but let's go ahead and talk about it in more detail here. So external respiration is what occurs at the interface between the pulmonary capillary membrane and the alveolar membrane. Essentially, this describes the process of gas exchange in the lungs. So we're picking up oxygen, we're getting rid of carbon dioxide. So here, oxygen diffuses freely across the membrane of the type 1 alveolar cells, across the membrane of the capillary endothelia. So because of the very high surface area, that diffusion happens very quickly. Oxygen will enter from the interstitial fluid, from the blood plasma into a red blood cell where it will be picked up by hemoglobin. And in that process of oxygen binding to the heme in one of those globin subunits, that is going to kick off a carbon dioxide molecule that itself was already bound to hemoglobin as carbaminohemoglobin. So what is happening there is the oxygen poor blood that enters through the pulmonary arteriole. That oxygen poor blood has a high percentage of hemoglobin that is maintained as carbaminohemoglobin. It's basically carrying carbon dioxide. So when oxygen binds, that decreases the affinity that hemoglobin has for carbon dioxide, so it kicks it off. We reform carbon dioxide by carbonic anhydrase, and that carbon dioxide is able to diffuse into the alveolar air. And then for internal respiration, this is describing the gas exchange that occurs in the systemic circuit. So here, it's going to be just the opposite. Oxygen that is carried by hemoglobin is going to respond to a great influx of carbon dioxide, which carbon dioxide will bind to the hemoglobin, decrease the affinity hemoglobin has for oxygen, and the oxygen will now be free to dissociate from hemoglobin and diffuse into the systemic tissues. So regardless of whether we're talking about internal or external respiration, the ability of carbon dioxide to convert from the gaseous form to the acid form by carbonic anhydrase is extremely important. So a good way to think about it is like this. Hemoglobin goes through periods where it either likes to bind to oxygen or likes to bind to carbon dioxide. Which status it is in is going to depend on how much carbon dioxide there is present, and specifically what is the pH of the blood. So we're going to go ahead and start working towards talking about that. So I've already shown you this figure from chapter 18, so I'm not really going to belabor it again, but again, don't forget the difference between the Po2, which is just the oxygen saturated in the blood plasma, and the oxygen that is capable of being bound to hemoglobin. So they're not really the same thing, but of course, hopefully you understand the vast majority of our oxygen that is carried in the blood is carried by hemoglobin. Okay, so let's talk a little bit more about the ability of hemoglobin to bind to and to let go of oxygen. So we're aware by now that hemoglobin goes through periods in which it binds to oxygen and becomes oxyhemoglobin, and then it goes through periods where it lets go of oxygen to deliver it and becomes deoxyhemoglobin. So this dynamic can be described graphically, as you see here, by what is called a dissociation curve. Essentially what you're seeing here is, on the x-axis, the partial pressure of oxygen in the blood plasma, which you understand is usually about 100 millimeters mercury in the arteries, or 40 millimeters mercury in the veins. And then on the y-axis, you can see the percentage of total hemoglobin complexes that are fully loaded up with oxygen. So one thing that we said is that in arterial blood, in which our Po2 is about 100, you can see that pretty close to 100% of hemoglobin is loaded up with oxygen. That's not really a surprise. But when it gets to deoxygenated blood, in which the partial pressure is more like 40 millimeters mercury, you can see that a significant percentage of hemoglobin starts to unload its oxygen. So instead of having close to 100%, it's more like 70%, which still seems like a lot, but it really is not that much when you compare it to 97%. So let's talk a little bit about the effect the blood plasma pH has on the ability of hemoglobin to carry oxygen. So this is all going to have to do with carbon dioxide, how much carbon dioxide is there, the effect that has on the pH, and the affinity that hemoglobin is going to have for oxygen. So this is going to be very important for us. So you recall that the set point pH of the blood plasma is 7.4. So what we have not really covered, even though we talked about why hyperventilation is going to cause the pH to go down and why hypoventilation will cause it to go up. But what we haven't covered is why in the world do we care? What is the big deal? So let's take a look at this. So what you're looking at here are dissociation curves for oxygen on hemoglobin at 3 different pH values. The purple graph at pH 7.4 is what it's supposed to look like all the time, right? At pH 7.6, this is what the dissociation curve might look like if we are hypoventilating, not getting enough carbon dioxide, hyperventilating, all right, I'm getting confused here. So at pH 7.6, let me walk myself through this because even I get confused on these things. At a pH of 7.6, not enough hydrogen ions, meaning not enough carbon dioxide. So if we are hyperventilating and getting too much carbon dioxide out, you might expect the blood plasma to be around 7.6. If we are hypoventilating and getting, okay, I think I've confused myself again. So if we are, let me just start with hypoventilating. If we are hypoventilating, we are not breathing enough, we are keeping too much carbon dioxide in, so that's going to lower the pH. So in blue here, you might see with hypoventilating. I think I got that right. So hyperventilating at 7.6, hypoventilating at 7.2. So if you look at these dissociation graphs, the pink one or the purple one right here is what things are supposed to look like all the time. The way dissociation graphs basically work is if you move the graph to the left, that means that whatever is doing the binding here, as in hemoglobin, the affinity starts to go up. So as the pH starts to go down, or starts to go up as things become more basic, hemoglobin basically gets to the point where it starts to grab onto oxygen more tightly. So if my phone here is my oxygen and I'm hemoglobin, so if things get more and more basic in the blood, I have a greater inclination to hold on to oxygen even more tightly than I already was. And that might sound like a really, really good deal, but it really isn't. Because, again, think about what has to happen in the life cycle of hemoglobin making its way through the circulation. Now, if hemoglobin is not bound to oxygen and it makes its way into the pulmonary capillaries, yeah, that sounds like a good deal. You want it to be able to grab the oxygen from the alveolar air and bind to it very tightly. But think about what would happen as you make your way from the left ventricle out through the aorta and into the systemic capillaries. If hemoglobin is holding on to oxygen too tightly, what is not going to happen? You are not going to be able to deliver that oxygen very effectively, because rather than let go of the oxygen and deliver it, the hemoglobin is going to want to hold on to it too tightly. And then for hypoventilation, when things become too acidic, it's just the opposite. As the dissociation curve shifts to the right, the affinity hemoglobin has for oxygen goes down. It's going to be way too inclined to give the oxygen up. It's not going to be able to hold on to it tightly enough to carry it. It's going to give it up way too easily. So it's not going to bind it very well when it makes its way into the pulmonary circuit. So the idea here is that if the pH of the blood becomes too acidic, hemoglobin's binding to oxygen is going to be too weak. And if the pH becomes too basic, the binding of oxygen by hemoglobin will be too strong, and we're not going to deliver it very well. But if you consider a pH of exactly 7.4, basically kind of think of it as a Goldilocks scenario. So this porridge is too hot, this porridge is too cold. At a pH of 7.4, the porridge is just right. This allows hemoglobin to bind oxygen exactly strong enough to carry it around, but not so strong that it can't let go of it when it's time to deliver it. It's just right, or as Thanos would say, it is perfectly balanced. Hey, remember what it was like to go to the movie theater? Neither do I, right? So, okay, so that will conclude this chapter on the respiratory system. We only have one more chapter left in this semester as we're talking in April of 2020. Might be different in later semesters, so if you're watching this much later on, we may go beyond chapter 25 on the urinary system, but if you are a spring 2020 student, the urinary system will be the last chapter that we cover. So I will go ahead and log off for the time being, and I will see you next time.